So we'll continue from where Henrik left off and talk about how to process and do quality control of the data once you've done the library prep. I will first take a bit of a detour and talk a bit about transcription and transcriptional bursting, then shortly about the mapping pipelines. And then I will go through all the steps that Henrik talked about and talk about what different issues we might have in our data and how we can deal with it in our computational pipelines. Shortly about single nuclei versus single cell and a bit about spike-ins, and then finally a bit on how we actually do the filtering and quality controls. So, transcriptional bursting. When you think about gene expression, you always think about a gene as being on and off, on or off. But when you look at the single cell level, a gene will be turned on and it will start producing mRNA. And then later on, that mRNA will be translated into protein. But then the gene is turned off and slowly the mRNA is degraded. Often more slowly, the protein is degraded. And we get this sort of pattern of on and off. And if we follow one, in this case, it's like an artificial reporter system with a fluorescent protein and a gene they can control. Each spike here is an instance where the gene is turned on to produce mRNA. At each time it's turned on, it will produce different amount of mRNA molecules depending on how long time it's on. So you might have a burst with just producing one molecule, but quite often when a gene is turned on and you recruit the machinery, it will circle around the gene and produce multiple copies of RNA. And if the bursts are frequent, you will accumulate and have high amount of mRNAs. If they're more if it's a higher distance between the peaks, then you will have time points like here where we have no copy of the mRNA. And of course, then the protein will follow a similar pattern, but more smoothed out, more or less, that it will never really go down to zero. And when we then do single cell sequencing, let's say we have a population of cells, they're all the same cell type, and we sequence a cell here, and we sequence a cell here, we will have quite high amount and zero amount of the same gene, even though it's the same cell population that has this gene as one of its classical marker genes. You might catch it at a time point where there's no mRNA copy of that gene. So that's really important to keep in mind when you're analyzing this kind of data, that a zero doesn't have to mean that the gene is not there for that cell type when you're looking at one single cell. And then, of course, with having these different kinetics of activation and inactivation of the genes, we will end up with these kind of different profile. A gene that's more or less constantly on, we will have all cells having expression of that gene. If it's sort of more balanced on and off, it will look something like this. Half of the cells will have it, half not. But we might also have, and quite a lot of interesting genes might have this kind of pattern where we see expression in very few cells. So we have this issue with bursting, but then we also have an issue with dropouts. So the step of reverse transcription, when we take the mRNAs and convert them to cDNA, will be a more or less random selection then of these mRNAs. But depending on the method we're using, there's no really good estimates, but I would say probably the best methods now are catching 40% of the mRNAs we have per cell. So this is what I would call a dropout, while the zeros we get from the bursting is actually just due to this stochastic zeros of the stochastic gene expression. If we do bulk RNA-seq, let's pretend we have this population. They're all the same cell type, and they only have 4 genes. With stochastic gene expression, they still look different. But if we do a bulk and take the average of that, we still get a good overview of what the common signal of that cell type. But then if we dissociate, we do reverse transcription, we do some more or less random selection, and then we do amplification, that it can also have bias due to like the length of the gene, GC content, structure, like RNA structure, and so on. So what we end up with after amplification will have a lot of biases. UMIs will help with removing the amplification bias to some extent, but not this bias. So you often hear when people talk about single cell data that it's zero inflated, and there's been a lot of papers and a lot of debates if this is really true, so I'm not going to state anything, but don't only think about the zeros as something we need to impute that are not only dropouts. There is biological information in these zeros also. So it's good to keep in mind. So when we have single cell data, it is much trickier to work with than bulk RNA-seq. We have this amplification bias, dropout rates, stochastic gene expression. We often have a lot of background noise. We have a bias in cell cycle, cell size, and so on, and often clear batch effects. So the correlation of 2 cells from the same cell type might look something like this, not a straight line like it should be in bulk RNA-seq. But the big advantage with single cell is that we have a lot of technical replicates since we have a lot of cells in most cases. Is there any questions on this part for now? Yes.
Thank you so much. I was asking, like in bulk RNA-seq analysis, like sometimes you have a threshold for the count matrix, like row sum greater than X. And in single cells, so we have a lot of cells which can serve as replicates. Is there also a cutoff to kind of get rid of low count reads, and if we do that, is it like good or bad? Because then like some genes are very lowly expressed in the sample per se. So maybe
Yes.
some cells catch up, some cells don't catch up. So how do we decide what to do?
We'll come back to this a bit later, but at the first step, we always do some filtering if it's not detected in 5 cells or 10 cells or whatever, depending on how big your set of cells is. It's normally what you always do, just to reduce the size of the data set you're working with and save some memory. But then we'll do additional filter later on. There was someone else had a question also? Yes.
Yeah, that was me. So these random selections of mRNAs, why is that? Is it like a limitant in the reaction of polymerase or something? Or
no, I mean, I would say that like the success of poly-A selection, it's not 100% random. Exactly which mRNA molecules will be amplified or will be reverse transcribed. It can have to do with the length of the poly-A or like the RNA structure. So it's not completely random. If you would count absolute numbers of molecules versus how much you get. That's the same in bulk RNA-seq. It's not completely like a one-to-one relationship there either, right?
Okay, thank you. And how is this known? Because of hexamer sequencing, or how is it known that not all of them are transcribed?
I would say most of them are transcribed, but some will be transcribed more easily. Like the chance of detecting them will be slightly higher for some than others. It's not.
Everyone gets the poly DT primer, but not everyone gets. Okay.
Yeah, I mean, that could be, but there can also be like the structure of the poly A tail. Yeah, there's a lot of tricks going into it. There's also actually what is a bit unclear exactly why, but it's known that we have a lot of internal priming that is not at the poly A end. We actually do get reads from like within introns and everything with 10x data especially. So not really sure why, and that is not completely random either. So
yeah, thank you.
Yep. So now on to the pipelines. Basically, after running this library prep, doing all these fragmentations and library prep steps that Henrik talked about, we take it to the sequencer, we end up with FASTQ file. And then we want to map it to some sort of reference to say something about gene expression. And the common thing, if you work with mouse or human data, is that you have a genome of the species you're working on. And the genome is annotated, so we know where the genes are. And then basically we just count how many reads do we have at each position in the genes, and we get a matrix with gene level counts. We can also get transcript level counts. So for each isoform, it's a bit shaky and the data is noisy enough as it is with single cell data. So I would recommend to work with gene level counts and not try to analyze each isoform. You can perhaps do that later if it's a special interest, but for clustering and so on, stick to gene level counts to start with. To not have even more noise. If we don't have a genome, we can also map reads to just the transcriptome. If someone has done the transcriptome assembly with these different pseudoliners, we can get transcript level or gene level counts also from that. But I will come back again now to the UMIs. Henrik explained it quite well, and we had some discussion, but just to clarify, in each of these droplets, we have this bead, and the bead contains all reads that is attached to the bead contain the same cell barcode, but also this random 8 base pair UMI. And then we lyse the cell, all the RNAs in this droplet will, or most of them will attach to the bead, and then they will be reverse transcribed. And we will have something like this. And then, or in another cell, we might have something like this. Then we sequence it, and computationally, we look at the combination of the UMI and what goes into the gene. And we reduce all the ones that have the same combination of UMI and transcript into one single count of that gene. So any sort of amplification bias is removed by using the UMI, but not anything to do with like the sampling bias. But it removes one source of noise in the data at least, and that's super useful. And since 10x is the most used method that a lot of you are using, and we'll have a quick look at their pipeline, just very much an overview. So the first step is just looking at the reads, if they even have the proper barcodes that we need, and throwing away those. Then we map it to the genome. Any read that doesn't match the genome or has a low mapping quality is discarded. Then we look at exonic intronic proportions. You can choose in the pipeline to only look at exonic or include also the intronic. And actually, with the later versions of the pipeline, it's default to include intronic reads also, so it's good to be aware of that. Intergenic reads are thrown out. Then it's checking again, antisense reads or reads mapping to more than one gene is discarded. Then we come to this part where we compare the gene and the UMI together to get these counts of UMI, barcode, and gene being unique in a combination. And then we remove all the duplicates of that same combination, and we have our UMI counts. Then we do the cell calling, which is basically checking which of the barcodes is actually cells and which of the barcodes is just an empty droplet. And we get our filtered matrix to work with later on. When it comes to cell calling, there are multiple different ways. I will talk a bit more about that later also. But normally, you will see these barcode rank plots in the Cell Ranger output. And then basically, you sort all your droplets on the number of UMIs it contains. So we have up here, high number of UMIs. Then we have sort of this inflection point here. And then we have sort of the background. So also the empty droplets will contain some RNA, some background RNAs, and will have counts. So if you have a nice experiment, it should look like this. It's easy to say, like, here I draw the cutoff, and everything up here is cells, everything down there is empty droplets. There's different ways on how to define this inflection points, depending on how you plot them, but the idea is all the same to find this point. Let's see. Oh, I forgot to make a copy of this. I just want to show you a report as an example. So here is one example of one of these barcode rank plots. So if you ran 10 experiments, you probably saw them. It just defines the inflection point here. You get some stats on like number of cells, reads per cells, mapping stats. I think it's important to look at sequencing saturation, for instance, because that, like we talked about with Henrik also, the amount of cells you load versus how much sequencing you get per cell, you can get a good feeling for with this. Do you have a question?
Yes, Åsa, sorry for interrupting.
Yeah, no worries.
Can you try to explain A plus V, the inflection point in this knee plot?
Yes. So you're clear on that. Here we have all the barcodes sorted by size, right? And here we have the UMIs. And we want to find sort of where we go from true cells to just background noise in this plot. And in this case, I mean, I think Cell Ranger has some strict cutoffs also on just number of cells.
Yeah, but what I'm wondering mostly is why is the inflection point in the middle of this drop? I mean, why is the cutoff set up there?
Oh, that's a good question. Probably to make sure, I mean, if we go down here, we probably include, but it would be fine to put it here also. There are other methods to put them at the knee, as they call it, or at the inflection. So this is not really clear. I would say it's better to include a bit too much that you're not certain about and do later filtering than to put it too high up here and remove something interesting.
Okay.
Did that answer your question?
Yeah, it's always tricky to explain to people why is the cutoff there, and then but it seems that it's the same for everyone. And then with Cell Ranger, you can force cells, right? And then you can increase
think
that
also we'll
probability.
talk more about background RNAs, and I think you should take that into account more also when you do this calling. I will come to that. Yeah.
Yeah, thank you.
Otherwise, I would say, I mean, 10x does just this calling, and then a quick analysis also with doing this t-SNE clustering, some differential expression, and so on. But you should always consider that this needs extra quality control and filtering, that this is probably not your final clustering. Also, I would say sequencing saturation does matter, especially if you're doing multiple samples and you have very different number of cells. You should aim to have sort of the same sequencing saturation in all of your samples. It makes it much easier to normalize it later on, that you have sort of about the same amount of reads per cell in all your samples that you're trying to analyze together. Let's see, I'll get back here. So now I will go through again these different steps that Henrik talked about. And I would talk about everything that can go wrong, just to make you more worried. I would say this is the dissociation is the most critical step to have good single cell data. If you're working with stuff with immune cells or anything blood that is not needed to dissociate, you're lucky, then you can avoid this. But otherwise, if you're working with solid tissues, you have to treat the cells with these digestion enzymes. Some of them will break the cells, and if the cells are broken, you get all this RNA leaking out that gives you a background signal. What you also need to be aware of is that different cell types will dissociate more easily than others. Some will break in the dissociation, so you will also have a biased composition of cell types compared to what is actually there in the full tissue. There's also this issue that if you have live cells and you add all these dissociation enzymes, you will introduce some transcriptional programs related to stress in the cells. So it's been shown in a few experiments that this specific dissociation signal is turned on in the cells when you dissociate them. And here is an example of the genes. Like I would say these UN and FOS genes you quite often see popping up in single cell data, and you have to be careful that it's not a biological thing between your cells, perhaps, but it's a stress signal due to dissociation. There's always this possibility that you need to consider. And as we talked about, you might get RNA leaking out into the soup, so to say, and then if you have RNA in your cell suspension liquid, it will end up in every droplet that you're running. So here is one example from a colleague that worked with data from 3 different time points. And as you can see in time point 3 here, we have neutrophils up here, but also all other cell types that are not neutrophils seem to have low expression of these several neutrophil markers. So it looks like we have leakage and broken neutrophil at that time point also. And then, of course, if we then go in and want to do differential expression of T cells from time point 3 versus time point 0, then it will look like T cells at time point 3 has neutrophil genes turned on. So you need to be really cautious and at least look at if you have ambient background RNA.
I was wondering also on the cell dissociation, if you look at metabolism, for example, could you still look at different metabolic genes, cell type compared to cell type, or would the cell dissociation like mess up this analysis that you can do? Because I can imagine this protocol itself already affects a lot the metabolism of the cells. So are you still able to do this sort of analysis then?
I would say yes, in most cases. I mean, there will be a slight influence of this background signal. And I think it's more when you have clearly distinct cell types and so on, that signal is still so much stronger. So if you're comparing across different cell types, it might be more an issue if you're comparing, let's say then, for instance, T cells between two different conditions. And one of the samples you're looking at, you did a different dissociation than you did in the other, so you have more of this dissociation stress, or it took longer time for your dissociation in one experiment than the other, then you might have issues to analyze the signals, I would say.
Okay.
Clear
Thank
enough?
you. Yes, thanks.
One super nice paper that is looking at this with ambient RNA is this paper where they mixed 3 different mouse strains and then pooled them together, did the sequencing. Then for each cell, they look at the different SNPs that are known for the different strains, and they define then each cell as belonging to either of the 3 strains, depending on the majority of the SNPs, and then they estimate the background based on all other SNPs that are from the other 2 strains. So basically, you would see that these clear cells that have a lot of counts from one of the strains, they still have a few reads here. That's the background from the other strains. But then as you go lower and lower in the numbers here, the proportion of this background contamination is higher. And actually, they did this with 3 rounds of single cell and then 2 rounds of single nuclei, so nuclei 2 and single cell 2. And these number 3 is the same sample, but with nuclei or single cell. And actually, in their nuclei experiments, they have very high background RNA, especially in this sample. So I would say if you end up with something similar to this, there's really no point in even analyzing the data when half of what you have in your cells is background. It's super hard to remove that. But if we want to remove this background, there's several different methods that are trying to do it. I would just mention quickly some of them. SoupX and DecontX are R-based tools that depends on first doing clustering of your data and then defining sort of your cell type signal and also what you have in your empty droplets and combining that to predict and remove a background signal. Cellbender is another. Didn't I have slides with that? No, I didn't. Is using a bit of more machine learning approach, trying to train on the empty droplets and predict and remove that. And they actually evaluated that on this data in this mouse mixed strains study. The problem is that we can't really draw any strong conclusions when it comes to any of these methods, because just from these, they're all kidney samples with the same sort of cells, but different methods have the largest error and the best scores. So, but in general, I think they say Cellbender behaves slightly better in these tests to remove this ambient RNA. So you can run that. You also get these sort of statistics on the amount of background per cell that you can use. And it also actually runs this barcode rank plot. I should have included a picture, I realize. When it does, I'll go back to the here, it calculates the background, and then checks the significant deviance from the background signal instead of just cutting on number of UMIs. So it can actually end up that you have some cells down here that are not a lot of background, and you might have some up here that are mainly just background genes that will not be called cells. I should add a slide with that. Yes, and then, as I said, we might have a biased cell type distribution. In this case, it's actually just comparing if you check fresh versus cryopreserved. Then we can see that some cell types, they survive the freeze and thaw cycle much better than other cell types that are becoming more or less depleted after freezing. But that's exactly the same with different dissociation protocols also, that you might get a biased cell type selection depending on what dissociation protocol you're using. So that was all about dissociation and the issues that can arise from dissociation. Then when it comes to capture, I would say the biggest issue is that we might have duplicates and empty wells. And also, depending on what method you're using, there will be more or less of a bias for cell size. It's known that 10x, like droplet selection, has a bias against really large cell types. But PARS claims that they can handle pretty much any cell size and has less of a bias. I don't know if that's true. There might also be some other biases depending on what method you're using. Also, what you need to keep in mind is the time. So the time of really going from live cells to dissociation and reverse transcription matters a lot for the quality, so that the RNA doesn't have time to degrade. But as we talked about before, any method will give doublets. Also, FACS sorting will give doublets, but not at all at the same amount. With 10x, as it is just a limited dilution, you flow in like your droplets in one direction and your cells in another direction. And it's just more or less a stochastic process where the more cells you load, the higher the proportion of doublets will be. And it's a linear increase. And they've done some experiment with like mixed samples and so on to estimate the doublet rates for their different protocols that you can have a look at their website. But in this case, it's like a very clear example with human and mouse. Most of the cells have either only human or mouse mapping, and then you have some cells in the middle that are mapping to both species. But it's not always that simple. This is another, it's one project that I worked in where I did my own doublet prediction. And I saw these samples here that looked like we have a lot of doublets. And then I talked to the group and they're like, oh yeah, these two samples, we thought them, but then the 10x machine was not working. So we put them in the freezer again and came back next week. And then, so these samples went to 2 freeze cycles. And I think probably we just have like cells sticking together, cell debris and stuff that looks like a doublet signal. And then also this sample was just lower quality. So these numbers that 10x gives you on how many cells you load and how much doublets you end up with is in an ideal experiment, but it might look like you have much more than that in your real data. When you look at the doublets in a UMAP, it's quite often these kind of streaks where you can see clearly that they sort of fall outside their sort of mix of this and that cell type or this and that cell type, and they fall sort of in between in the UMAP. But I think there was someone who asked at Hendrik's lecture about this with the doublets. I don't remember exactly what was the question.
Yes, my question was because sometimes like doublets, like in the recent papers, it's like not only like they are technical issues, but they can be like signs of being co-expressed, or since they might be having a communication together. So this is the reason they stick together. So then how do we segregate from technical doublets to real biological doublets? Or should we always remove them first and then go
I
back
would
to the
say
data?
always remove them, but of course, I mean, what you can do is just do statistics on of the doublets. I find how many of them have this plus this cell type signal, and thereby you can get is, and then you have to really go back to statistic and say, I have 20% of this cell type, 10% of this cell type. I get this many doublets of those two cell types, and is that likely just by random, or is it enriched more than I would expect by random? That's the only way to say if I have more doublets of specific cell types. Another big issue with doublets is if you want to do trajectory. So in this project, we do have a transition of cells going from this cell type to this cell type. And they were super interested in studying this transition of these two cell types. Now, it's super tricky to see where do I have the doublets and where do I have cells that are sort of half in between two cell types. But actually, we managed to try to find stuff that has more signal than just a mix of two cell types. But this will become super tricky. So if you want to do trajectory analysis and that's your main goal with single cell data, don't overload, because doublets will be a big issue for trajectory analysis.
Is there
And
also, sorry, can I ask another question, please?
yeah,
Is there a minimum cutoff, like from like I have this many doublets, I have 10% doublets, so that's okay for my data to be analyzed? Or if it's more than 20% or something, then it's a bad data quality?
it's not so easy to say because it depends a bit on what exactly you're planning to do, what kind of analysis you're planning to do. For instance, for trajectory, I would say 5% doublets is too much, for sure. But if you're just wanting to define the main cell types and do some differential expression or something, then maybe it's okay to have some doublets. So there's not one simple answer.
Thank you.
I had a question actually, because you mentioned that if you have these like streaks in the UMAP, then most likely that's due to doublets. So my question is, in a data set that I worked with before, there were two clusters that were defined, and one of them was kind of on one and the other side of the second cluster. So my question is that also a sign of doublets or anything that's bad with the quality of the data itself?
Could be, but it needs more exploration, basically. I mean, it's hard to say just from looking at the UMAP. So always I would also look at, if I have these cells here and I have these cells here, and then I look at these cells here, they have markers of both cell types.
Mm-hmm.
Or if they have like these low quality cell signatures and so on. So it always takes some detective work, basically.
Yeah. Okay, thank you.
Yeah. So then, of course, if we have doublets, we want to detect them. We want to remove them. If we are sequencing just one cell type, then sometimes you can just take anything with too many genes detected is most likely more than one cell. But that doesn't work so well if we have mixed cell types that are of different size and different RNA amounts. So then, I mean, we can check manually for clusters with multiple signals and so on. But it's probably better to use one of these prediction methods. So we have many methods. These are some of the main ones, but their idea is more or less the same. We have our data. We make simulated doublets by randomly taking pairs of cells, adding up all the counts for them, putting that back into data, rerunning PCA. And then for each cell we have in the data, we count how often does it group together with these simulated doublets, define some sort of cutoff, and then remove those that we think look like doublets. And then exactly what algorithms and how they define these cutoffs and so on are, that differs a bit, but the idea is the same. But still the results are slightly different, so maybe it's a good idea to run 2 different tools and compare. Yes?
Yes, thank you. Isn't it just easier to plot UMIs versus counts? And then...
Ooh, if you have one cell type, that works well, I would say.
But
if you're only sequencing T cells, for instance, and they're not cycling cells also, would help, because it's not as easy as having more UMIs just means that you have a doublet. Because if you have a doublet of 2 small cells, that's still a doublet.
Yeah.
But it will not have a large UMI count.
Okay.
So what you should be aware of is that we can detect doublets between cell types. So a doublet of a B and a T cell, or a fibroblast and a T cell, we can detect with these kind of methods. But a doublet of the same cell type, we can, I'm not aware of any method that can detect it, because there it's mainly just having higher counts, right? That's the only way we can say.
Yeah, but if you plot it against the UMIs, then you get a diagonal.
Yeah.
Right? And the diagonal is the one that is real singlets, and the ones that fall up above or up the diagonal, they are doublets. Because they have, they might have more RNA, but not more UMIs, and the other way around.
It would be super nice if it was that simple, but it's the cells are not that uniform, and the library preps are not that uniform, unfortunately.
Okay, but then in Seurat, the first step is actually to remove doublets used by checking the number of counts.
Yeah, in some of their tutorials, but not all of them.
Okay.
But that's not what I would recommend to do.
Okay, thanks.
Yeah. Okay, then the next step in the analysis is the lysis, and that is, of course, we might get breakage of some cells and some might not break. What you can notice is that you might get more or less of a nuclear bias in different cell types, that if you don't lyse the nucleus of all cells, that will bias your RNA content. Otherwise, I think this is fairly straightforward part. Then, of course, at the reverse transcription, we lose a lot of the transcripts due to dropouts. It's also been shown in several experiments that for some reason, even though we have all our cells in different droplets, some droplets will have better efficiency in reverse transcription than other droplets, or some wells if we're doing any of these well-based methods. Really unclear why, but the reverse transcription efficiency seems to vary from library prep to prep. When it comes to the amplification, we do have a bias, we know it. UMI controls for it to some extent, but of course, the chance of detecting a transcript is higher if it gets amplified more. And then, of course, if we don't have complete sequencing saturation, there is still a slight bias due to amplification also with UMIs. And then, of course, there are some methods that do not have UMIs. And then sequencing, most likely one of the minor contributors to any sort of technical artifacts, but it's still good to think about how you pool your samples, and if there's a split on multiple lanes, that there might be some bias. It's also been shown that there is some index swapping between the cells when we sequence. But mainly, I would say the biggest contribution to bias and so on is from the cell dissociation step and single cell capture. That you really need to optimize and think through how you're doing things to get the best quality cells. So to sum up, if we're thinking droplet based, we have droplets with the cell and the barcode. We have a lot of these empty droplets. We might have droplets with dying cells. We might have droplets with ambient RNA or with cells and doublets. And in the end, we want to clean this up and only keep these ones and remove everything else and have a nice clean matrix to work in. But first, I just want to mention this with the single nuclei. So we can do single nuclei instead of single cell if we have tissues that are hard to dissociate. For instance, neurons with their long axons is really hard to get whole cells. So our only option is to do single nuclei instead. Adipocytes, or if we have frozen tissue, that will break the cells if we dissociate it. So, of course, we have less mRNA in a nuclei than we have in a cell. We get much more of these nuclear link RNAs. We get a lot of these intron structures, and we also get more ambient RNA with nuclei. So if we can get cells, it's better. But if we have a tissue where we can't get cells, nuclei still works. There's also been shown that some cell types are harder to actually get the nuclei from. For instance, microglia is one example where when you do nuclei dissociation, you also get a biased gene selection, like gene program in the microglia versus if you do cells. So nuclei data will look something like this, that we have intronic reads versus exonic reads higher in cells, intronic higher in nuclei, slightly more intergenic, more total reads, more genes detected. And also, if we look at pairwise correlations with similar cells, you see that it's clearly shifted down if we do nuclei of the same cell types. And that you also make sure that you're also counting intronic reads, since we have a lot more pre-mRNAs in the nuclei data. Spike-ins are mainly just used for SmartSeq data. So I would just mention that they're there. It's not possible to use spike-ins with droplet-based assays. The whole point of adding them is like an external source of RNA that we can use to control for technical noise. So to check for amplification bias, dropout rates, use for normalization and so on. But I think I will skip that part for now. So back to this picture again. We know that we have all these issues. We know that we have dying cells, we have empty cells and so on. We talked a bit about different methods for doublet removal, and I would suggest you run them. We talked about ambient RNA, and it's a good idea at least to look for it. There is some sort of warning in Cell Ranger if it thinks that you have a lot of background signal, but it doesn't really tell you what it is. Also to compare if you're running multiple samples, if your background signal is the same across samples, it's probably fine. But then when it comes to filtering sort of the quality of the cells, we'll have a lot of these low quality dying cells and so on that we want to remove. There's a lot of these different stats. Now I put a long list of stuff that people are using to filter, but I will go through them. If we start with just number of reads, and also mapping statistics. That's mainly for full-length methods because we can't get that for 10x data, proportion mapping to exon, proportion mapping to protein codings or mRNA. All this tells us something about the quality of the data. If we have low number of RNAs going into a library prep, we will have less number of reads, less number of genes. We will also have more primer dimers and weird stuff in the library prep that will lead to weird mapping statistics and so on. Also, we'll have less mRNA mapping reads. So all of these together gives more or less the same. If we have spike-ins, we can also look at the proportion of the spike-ins that are detected, if it's a good library prep, also the amount of RNA we had to start with based on the ratio of spike-in to endogenous RNA. I really like to look at number of detected genes because that more than the number of UMIs tells us something about the complexity of the cells. And this is sort of a mock experiment where they sorted multiple cells in SmartSeq data in the same well versus a single cell, and then we had some clearly failed libraries. So just looking at the distribution, you can quite often see, like you said, the doublets having higher numbers and the failed libraries having lower number of genes. But it's tricky because these could be like two very distinct cell types having very different numbers of genes, but it shouldn't be this distinct, perhaps. But you have to be aware of that cell size and number of genes and spike-in ratio, all of these are correlated. So here is an experiment where they fax sorted the cells, and the forward scattering in the fax is supposed to be sort of a proxy for the size of the cell. And if we look at forward scattering versus spike-in ratio, you see that there's a clear correlation, even though it's not perfect. And also like number of detected genes versus spike-in ratio clearly correlated. So cell size and how much RNA we detect is clearly correlated. Then mitochondrial read fraction is something that most people are looking at for filtering. There are sort of two reasons why mitochondrial read fraction comes up. And it's been suggested, I don't know if it's been proven, but it's that when the plasma membrane breaks, that a lot of RNA is leaking out, but whatever RNA is in the mitochondria is more sort of intact and not degraded and lost. So then the proportion of mitochondrial reads will increase. But then also we know that a lot of mitochondrial signals might be due to apoptosis also, that it induces some mitochondrial transcripts. But it's been shown in several experiments with like leaving the cells on the bench for an hour and so on, that the mitochondrial signal really increases with the quality, with low quality cells, and it's clearly related to quality. Also, ribosomal proportions. There has also been some studies showing that the proportion of ribosomal proteins will vary with like handling of the samples and freeze-thaw cycles and so on. When it comes to ribosomal RNA, it's something you can see if you have really degraded RNA, that also the ribosomal RNA is degraded, and you start getting templating and reads from the ribosomal RNA. That's really an indication of bad quality. And then if you have full length, we can also look at the RNA degradation based on the 3' bias. So basically, if we have, since we're catching the poly A tail, if the molecules are broken, then we only end up with reads close to the poly A tail and not covering the full transcript. So that's also a bad sign. But I would say these 4 are the main things I would look at. And when it comes to mapping stats and spike in detection, that's only for SmartSeq data. So when it comes to 10x data, for instance, I would mainly look at these 2 always, and then the other ones sometimes, depending on what I have. But for sure, always number of detected genes and mitochondrial read fraction. Then how do we filter? I would say look at the distributions. That's my best suggestion. To find stuff that deviates, like if we have the mapping stats, I see this tail, I remove it here, or I have number of detected genes. Here I probably have my doublets and my failed libraries. So just visualizing the data and deciding cutoff is probably the best way. We can try to do some automated detection based on the distributions and so on, but you should always look at it also. Then, of course, there are some tools that are trying to take these QC metrics and clustering on the QC metrics and detect outlier cells. But I haven't used them so much, I can't say much. There are a couple of new papers out that are suggesting a completely new approach to this quality filtering, and that's to do some sort of rough clustering first. I think there's a good argument for this, but I haven't evaluated it so much. But the whole idea is that if you have here number of detected genes versus percent mitochondria, and we have 3 cell populations that have very different pattern when it comes to these 2 quality stats. We might have failed blue cells ending up here, and we might have failed blue cells ending up here, but we don't want to set filters so that we remove all the red cells, for instance. So then instead they do a very rough filtering, then clustering, and then for each cluster, they remove everything that's sort of outside of their main distributions. But then also some extra round of QC, and then select a set of good quality cells. There's another paper doing more or less the same as this DDQC. I think it's super nice at least to think about these issues. Here they're showing the fraction mitochondria, number of genes, and fraction ribosomal for a bunch of different 10x experiments from different tissues and different labs and so on. And you see you have very different behavior for the different data sets. And if you take one data set, you split it up, do the clustering, you see that you have even more different behavior for the different cell types within one data set when it comes to some of these stats. And they do a similar thing where they first do basic QC, some clustering, and then calculate thresholds for each cluster. So this could be a way forward if you have very complex data with many cell types, to not just do one cutoff for the whole data set, but instead combine everything into one QC. So when it comes to filtering, how do we decide cutoffs? As I've mentioned, if we have one cell type, you expect more of a homogeneous profile for your whole distributions. You expect sort of the same number of genes, sort of the same proportion of mitochondrial reads and so on for all cells in your sample. And then you can do fairly strict filtering on those distributions. But if you have a very complex tissue with many different cell types, you should be quite careful in filtering too much, I would say. Especially now this with doublets. I've had experience where we have like all the tumor cells look like doublets if you just would do that filtering based on number of UMIs and so on. but they're actually the super interesting cells I want to study, but they have much more genes and counts. Does anyone have questions for now on these parts?
Yes, I'm just wondering about the ribosomal RNA. Do we include in the library prep step itself removal of the ribosomal RNA?
Yes, I think there is in... but no, this is polyA selection, so there isn't one. But there are some of these library prep methods that are based on random hexamers, and then they include one step for rRNA depletion.
Yes, so if this is a polyA selection, so in that case, do we still get some inference or interference from the ribosomal RNA?
The thing is that we get a lot of crap that's not polyA, basically. And we get internal priming in exons, we get weird stuff, we get intergenic reads, and what we've seen is also that like cells sitting in the freezer for too long, we get more of the ribosomal RNA. And I think it's probably that if the RNA breaks up, somehow we get templating in there. Not really sure why, but that's more from just seeing that we have it in the data.
Yeah. Thank you.
Yes, ambient RNA cannot be removed, right? We only know that it's there, but it is there. So if it's too high, better not to use that sample.
It can be removed. I mean, that's these methods. Where did I have that? Here are some methods, and here they are also evaluated on how well they work at removing. So I think if you're somewhere here with samples that have a bit of our background, I think it works fairly well to remove it. And it's also fine to work with it. Just, I think when you're comparing across samples, you have to be super cautious and know what your background signal is. And like if the differential expressed genes you get are the same as your background signal when you're comparing conditions, you should be worried. But if you're starting to be like 50% background, I would say throw away your samples.
Thank you.
Yeah.
Hello, I have one question for the cell dissociation. So, the cell dissociation method is important for all kind of cells?
Yeah. I mean, any cell that has is a solid tissue, right?
Yeah.
Then you need to dissociate it into single cell suspension before we can do any of these methods.
Okay, I mean, for immunocells also these are important?
No. I mean, immune cells, if you're looking at like tumor infiltrating immune cells, then you have to take your tumor, dissociate it to get out the T cells or whatever you're looking at. But if you're looking at blood immune cells,
Okay.
then you're fine. Then you don't need to dissociate.
Okay, thank you.
Yeah. So after we filter cells that we believe to be low quality or failed libraries or doublets, we might also want to filter genes. We had a question before on this with removal of genes. The most common thing is to say if it's not expressed in at least this many cells, sometimes also with a specific cutoff on how many counts. But the normal thing is for a gene to be included, it should be detected in at least 3 cells. But the more cells you have, the higher that number will be. But of course, then also if your smallest population of cells you have in your data set is maybe 20 cells, you don't want to go too high on those numbers. But that's also the main reason for doing this cutoff is just to reduce the size of your matrix and save some memory in your scripts and in your computational cost there. But we will also do specific selection of genes for analysis, like variable genes. We'll talk more about that in the normalization lecture. we might want to filter out genes that's not correlated to any other genes, or sometimes filter for genes that we know, like immune cell genes or differential expressed genes from bulk or top PCA. There's a lot of ways to select the genes that we analyze on. But when it comes to really removing and filtering, I would say plotting these top expressed genes is a super important thing. This is, in this case, SMART-seq data where we also have spike-ins. So we see these ERCC genes being the top. Here we have the median, and we also have per cell. So we have some cell where 30% of all the reads are made up of this transcript and so on. Sometimes we have these genes that make up a large proportion of the transcriptional landscape per cell, like more than 50% of the reads in many of the cells is from one single gene. Some of these known nuclear link RNAs pop up in this list. Also, mitochondrial genes are common to see. So if you see MALAT1, for instance, in both mouse and human, is one of these link RNAs that are often really high. And sometimes also like actin or hemoglobin genes and so on for specific cell types. There might be a need to filter out those genes just because they mess up our normalization too much. But also these nuclear link RNAs can actually be related to quality of the cells. It's sort of the same thinking as with mitochondrial genes, that the link RNAs in the nucleus is more protected against leakage and degradation than the ones in the cytoplasm. So if the plasma membrane is broken, you tend to get more of a nuclear signal than if your plasma membrane is intact. So I would say always remove the mitochondrial genes, because we know that so much of the mitochondrial signal is technically driven. So I always remove them. I look for other suspected technical bias and very highly expressed link RNAs, and possibly other really highly expressed genes. That is what I also pretty much always filter for. Some people are suggesting they should always remove the ribosomal genes. There are different opinions on this. You can do so if you want to. I sometimes also subset for only protein coding genes to not deal with link RNAs at all. And then I take that subset for analysis. Then also we will have a lot of batch effects. And then a lot of the issues we have, like ambient RNA, time of handling of your samples and so on, will give strong batch effects. And if you have patient data, you pretty much always have strong batch effects. We will talk more about that in the data integration lecture. But for now, I think it's important to think about that when you visualize your QC measures, look at them per batch. So for instance, here, if I have different dates that I picked my cells, or I have different date that I did the library prep on for different samples, I should look at all these different QC stats that I'm using with all kind of batches I can think of to see if there is something technical that I might need to deal with. And also always look in PCA for batch effects, and just be warned that with single cell data, pretty much always PC1 or 2 is driven by like number of detected genes, as you can see here. And you can often see clear batch effects just in the PCA, so it's good to explore there. So that sort of wraps up the QC. Start with filtering clearly failed libraries. It's better to not filter too strictly in the first round. Do some gene filtering, and then do the first parts of the analysis. Then visualize your QC stats per sample onto UMAP and so on, and see what issues you have. And maybe you see, I have this low quality cluster that pops up, or these streaks of doublets or whatever. And then you go back and you do even more filtering. And in the end, you end up going QC, visualizing your data, clustering. I need to tweak it some more. I go back to the quality control. And so it's better not to filter too much in the first round, because then you might remove an interesting cell population. It's better to have some bad clusters in your first round of analysis and then having to repeat it, I would say. So try to plan to avoid too many of these batch effects, and also think about speed a lot when you prep your single cell data so that you don't have degraded RNA and dying cells. When it comes to defining cutoffs, think about what you expect. Do you expect one single cell type with uniform distributions, or do you have homogeneous cell types? And to remove doublets, we can always run predictions, but we will never get rid of all doublets. So be a bit cautious about how many cells you load. Yes, that was it. Any questions on more on filtering and QC?
Yes, thank you. I'm just wondering about the filtering based on highly expressed genes. And you gave nicely an example of the MALAT one. However, so when you filter out, so do we filter out completely from the analysis, or we exclude it from the clustering? And
I would, slightly depending on how many genes and which genes. Sometimes I would exclude it completely from the analysis, especially if it's like super high, up to 60-80%, or I mean over 50%. Because it's also a big issue for normalization, that one gene at 50% will push everything else down when we normalize by total counts.
Yes. When you say 50%, you mean to say 50% of the total read counts or 50% of the cells?
50% of the total read counts in a single cell.
In a single cell. Oh, okay. Yeah, it makes sense. Yes. Yeah. Thank you.
Okay.
I have a question regarding, because many of the cancer cell lines that we transfect in the mouse brains, they have like these marker genes like GFP or TD tomato and so on. So how much, like by experience, do you see like they also pop up a lot, like in the top 38%? And then if we also use a spike in, how do we normalize something like this?
Okay, when it comes to this marker like GFP or other reporter genes, you will, of course, first you have to consider that you add them in when you do the mapping, so you actually have reads for them, then you can study how much of them you have. My feeling is that they're not usually among the top genes still. Never seen them pop up among the top genes in my experiments. And you should also be aware that, especially with GFP and so on, it's fairly stable protein, but the RNA is usually not that highly expressed, and you will have like the majority of the GFP sorted cells will not have the GFP transcript even. But the other question was,
if I have those genes in my read counts, like how do I normalize it with context with the spike-ins? Like,
yeah, I mean, I wouldn't recommend using the spike-ins for normalization. Basically, it's good for quality control to check the amount of RNA and the detection of them, but and possibly for modeling technical noise, but not more.
You said that when it comes down to mitochondrial RNA or genes, usually you have to filter off cells with high number of mitochondrial genes, but you also get rid of them after the filtering. You also get rid of the mitochondrial genes for further downstream analysis.
So first, I calculate the proportion mitochondrial, filter cells that are extremely high. Then I remove them from the matrix before running the normalization.
Good.
Because I believe it to be mainly technical, and it makes up still, I mean, a fairly big proportion of the whole transcriptome.
Yes, I just have a comment and a question regarding the GFP or the reporters, because these sequences, if we use either human or mouse cells, for example, they are not mapped already because they're actually exogenous. They don't exist in our genome. So this is excluded from the mapping.
No, you can include it. So in all these pipelines, when you run mapping, you just add these extra sequences.
Yes, that if you want, right? Yeah. But by default, if you don't add it, so it will be excluded. So it won't be a problem.
You need to add it.
Yes.
And I should mention this, that I know that if you sequence at NGI, for instance, and they run the pipeline for you, it's very important that you tell them that I have these extra sequences in that I want mapped.
Okay.
Because otherwise they run the default just mouse or human genome, whatever you said.
Yes.
But if you have additional sequences, they can add them in.
Okay. And what happens if we overexpress? We are using like established cell lines, okay, not cancer cell lines, and then we overexpress a certain transcript. So in that case, it will be expressed at a very high level. So do we exclude that again from the during the QC?
I mean, I would remove that gene before running normalization. I would store the value as a metadata thing, but probably not include it in the normalization. If I want to compare like cells with overexpression versus non-overexpression, it would make sense not to include it, I think.
Yes. Okay. Yeah, thank you.